name is Dorian Bruch, and I'm going to be a second-year chemical engineering major here at UCSB. My mentor is Dee Morales, and my faculty advisor is Dr. Norbert Reich from the Department of Chemistry and Biochemistry. And my project is titled Light-Controlled Protein Delivery Using Plasmonic Nanoparticles. So when we try to understand how a cellular process works, one of the best ways that we've come up with is to deliver a protein to a cell and see what its effect is on that cell. And so the methods of delivery are pretty well established. And so one of the most popular methods is you have a protein, and on it is a peptide called TAP. And this TAP allows it to be able to get into the cell. However, this method is very time consuming because it takes a long time for the protein to be able to get into the cell. And so when we try to study very timely processes, such as mitosis, we don't have time to sit there and wait for that protein to get across the membrane. And we need to be able to have it there active right away so we can study it properly. So scientists have been moving towards light control. And so one of the solutions that they have come up with is the use of something called an optogenetic protein, which is just a protein that is controlled by light. So essentially, in darkness, the protein is folded in such a way that it is inactive. But when you shine light on it, it refolds and becomes activated. And so that way, they're able to time when they want to activate the protein and where they want to activate it by shining light on it. However, designing these proteins, it requires substantial effort because it involves going and changing the individual amino acid sequences in such a way that it has this light activation, yet still maintains its structural and functional integrity. And so people spend entire PhD programs, years of research, designing one protein. So we can avoid that problem altogether and use nanoparticles. Because with a nanoparticle, you can get the same, you can attach a protein to its surface, you can tether it to it, and then you can shine light and release it. And you can use it for virtually any protein. So not only does the nanoparticle have the same spatial and temporal control as the optogenetic protein, but you can use it for virtually any protein to study many diverse cellular processes. And so my main goal is to be able to get fully functional protein into cells using nanoparticles with light control. And so I had to use a few methods to do that. The three main ones being protein purification, nanoparticle assembly, and then delivery and evaluation. So for protein purification, I need pure protein to be able to carry out my experiments. And the protein I was working with in specific for my project was P53. And in order to get this P53, I grow up some bacteria cells, and then I lyse them and get all my crude protein, which contains other proteins and some cellular junk. And then I purify it, and I get my crude protein. But how do I purify it? Well, there's this special chemical called NTA, and this NTA can claw onto a nickel ion. And then I have a column that's coated with this nickel NTA construct. That way when I run my crude protein through it, my crude protein has something on it called a polyhistidine tag, and this polyhistidine tag has a special affinity for the nickel ion. So when the crude protein runs through the column, it sticks to it, whereas everything else is supposed to run out. The next thing is, I needed to design a nanoparticle in such a way that it has light control, but it can also tether protein to it. And in order to get this light control, the tether itself is bound to the surface of a nanoparticle using a thiol bond, and this thiol bond is semi-covalent. So when I shine light, infrared radiation in specific, onto it, it breaks that bond off, and then the tether is released. The next part is in order to tether the protein to it, I use the same nickel NTA construct that is present in the protein purification in order to tether it on there. The last thing is when I deliver the P53, I need to have a way to be able to tell that I successfully delivered it. So in order to do that, I'm going to use, it's called the P53 promoter plasmid. And essentially what it does is there's two main features to it. The first main feature is this little red thing right here. It's a gene sequence called DS-Red. And what it is it codes for red fluorescent protein. And as that name suggests, it's a protein that fluoresces red under the microscope. The other important part of it is another sequence called P21. And all that P21 is in the presence of P53, P53 will bind to that sequence, and then it activates the expression of ds-reg. So that way, what I do is I take this plasmid and I transfect it into a human cell line, which just means I put it in there and it gets incorporated into the genome. So then once it's transfected, I can deliver P53 to it. It'll bind to, it should bind to the plasmid, and it will induce expression of red fluorescent protein. And I can look at it under a microscope and confirm that I delivered it. So what are my results? Well, when I first started this project, we had a lot of P53 stock protein, and I was going to try to avoid the idea of purification altogether. And so what I did was, if I was going to use a stock protein, I needed to make sure that it was active, fully functioning, and pure. And so what I did was I ran something called an EMSA, which stands for electrophoretic mobility shift assay, which is just a fancy way of saying an assay that determines whether or not your protein is active or not. And so I run this on a gel. And so for those of you who aren't familiar with a gel, you have your starting point where you load everything into the wells, and then where you stop it is where it finishes. And so the wells, they're hard to see, so I outline them for you. And additionally, when you run a gel, things are separated by size. So the bigger they are, they don't run as far down the gel. So essentially with this method is I have a fixed concentration of DNA throughout all the wells. And I have one where it's DNA alone, which is a control. And so what I did is I take my P53 and I incubate it with this DNA. And the specific DNA I'm using, the P53 binds to it. And so as I increase the concentration of P53 incubated with this DNA, there should be more and more binding. And as you can see right here, this band intensity decreases relative to the control. So you can see that there is more and more binding as the concentration increases, which suggests that the protein is active. However, since the protein binds to the DNA, I should be seeing a band somewhere around here that would be forming, which would be the DNA protein construct, because it's heavier than the DNA alone. However, it's not there. And after many gels and optimization, we finally concluded that you can see right here, there's actually this stuff that's forming at the top. And it increases, the amount of stuff forming at the top increases as this band intensity decreases. So what's actually going on is the protein is aggregating to the DNA, and it's so big that it's unable to run through the pores of the gel. And so what I did afterwards was I went ahead and I checked the purity of the protein stock I was using. And right here, this highlighted section is the P53 at 3 different concentrations. And you can see that because there are so many other really dark bands everywhere, that the protein is quite impure, and that is causing it to aggregate. And so I had to go through and purify it. And so that's what I did. Well, I did not purify it. And this seems like a lot of data that's right here, but I'm going to walk you guys through it. So with this purification, here's the ladder right here, and this allows me to gauge the size of the proteins so I know which proteins I'm looking at. And so the first well corresponds to the crude protein, which is just the protein that came straight out of the cells and was ran on the gel. So you can see that there's a lot of bands everywhere. There's a lot of different proteins. The next one corresponds to after it was loaded onto the column, it was washed with some buffer and stuff ran off. Because even though the protein is supposed to stick onto it, there are other things that stick onto it as well. And so this was washed, and so you can see some stuff came off. And then these next wells, I increased the concentration of a chemical that forces the protein off of the column, and that way I can collect it. But I do it in variable concentration, so I'm able to kick stuff I don't want on there first off. So you can see with the first concentration, there's a lot of stuff that came off. Not much here, but then there's stuff here and stuff here. And so you can see that there's actually a thick band forming right here, and that does indeed correspond to the P53 protein. So you can see that it is indeed getting purer and purer, and right here, it's not a 100% purity, but it's way better than what it was before. And so there's this band that is forming that is my P53. However, with this short 8 weeks I have for research, I was spending a lot of time on optimizing this process and all the other processes I did before that. And so I wasn't able to get to the delivery of the p53 using the nanoparticles yet, but I'm going to continue that once I come back to the fall. And hopefully, what I'm going to do when I get back, well, what I'll do when I get back is I'm going to establish control showing that that p53 promoter works, and then I'm going to complete the delivery. And so when I get back, I hope to be able to shed some light on the use of the nanoparticle technology when it comes to delivering proteins. And I'd like to acknowledge my mentor, Dean Morales, for mentoring me this summer and spending countless hours in the night in the lab. The Reich Laboratory for allowing me to do research in their lab, the California Nanosystems Institute, and the Centers for Science and Engineering Partnerships for funding me, and the Neural Research Institute for providing me with some equipment to use in this research process. Thank you for listening. Questions? Yeah.
So Dean, because your initial sample, I'm sorry, did I just call you Dean?
That's okay.
Sorry, Dorian. Let's start over. Dorian. Sorry, I'm saying Dean. Since you were saying that your initial samples that weren't very pure were causing a lot of problems, do you feel that the purity that you have now is sufficient to move forward, or are you still going to try and purify it further? Do you feel confident in your new purification protocol?
Yeah, we actually did another purification on, there was another purification that was done, and there was protein that is nearly 100% pure. It's pretty close. It was a really good purity. So I'm gonna use that, and so that'll be good. That'll work out. Yeah.
Is P53 commercially available?
Yeah. So you can purchase pure protein, right? But it's very expensive. It's like you get just a little bit, maybe like 100 milligrams or something for like $500, and like that doesn't go that far. Honestly, when you perform countless experiments, it's way too much money. This is like just $10. Okay. Yeah.
So you showed an image where the protein is bound to the nanoparticle by a carbon-sulfur bond, and you break that through light. So do you form radicals from that then?
You know what? It's the part of the bond that's broken is the sulfur bound to the gold on the surface. So that's the part that's broken.
Okay, so it's still light initiated. Is that still a radical that's being formed?
Yeah. So when the initial thing is formed, I believe there is a radical form, because when we synthesize these nanoparticles, you have something called PEG, which is polyethylene glycol, and it has this disulfide on it, and then you cleave that and it forms a radical, and then that's what binds to it. It's reformed. Yeah.
So if it works, you think your method will replace the optogenetics, which is so popular these days? Or in other words, can you think of a scenario that optogenetics still has its use?
Yeah, so we're trying to get this technology to have about a 60% efficiency. So it's still in the progress. And I think once we can get it at a 60% efficiency, then it will become robust enough to be able to be used extensively. And for the optogenetics, I know that one of their favorite proteins to use is J-alpha helix, and I'm not too familiar with what that protein does specifically, but I'm sure that with the wide variety of proteins that you can use to attach to the nanoparticles, that there would be things that you couldn't do with an optogenetic protein that you could do with these nanoparticles, because no one wants to go through and change up p53 in such a way that it has light activation. It's so much effort.
Right. But on the other hand, just for your reference, right, if you work with some transmembrane protein, right, then optogenetics has its advantage.
Yeah, definitely.
Because if you put a transmembrane protein directly into cell through nanoparticle, most likely will not work because they have to go through the secretory pathway, is that right? To be targeted to the right place.
Right.
So each method has its own advantage.
Definitely, and so this would provide alternatives to be able to further some other research. Yeah.
For the light activation, you said it basically like severs the bond between the nanoparticle and then the protein. Like roughly how long does that take for when you activate the light, and then what happens to the nanoparticle afterwards?
So what happens is it severs the bond, it cleaves the bond between the sulfur and it severs this bond right here between the gold and the sulfur. And so when that happens, the tether is released, and that happens extremely quickly. It's like on the picosecond scale, I believe, for it to sever. And then the nanoparticle just stays there. It's non-toxic. It doesn't kill cells. If anything, you're more worried about the nanoparticle heating up and killing the cells by heat. So it just stays there. And then this tether releases from the protein once it's all off too, because of the concentration shifts equilibrium in such a way that it comes off. So the tether doesn't interfere with anything either. Yeah.
So when the tether comes off, what quenches the radical? Like what does it because you have a free radical in the cell.
What it could do is it could possibly, I mean, I'm not so sure. I haven't thought about that in that way. I haven't talked about monitor, but if I had to think, there's other tethers in there and they're both radicals, so maybe they'll combine together. But I could ask my mentor to get be sure about it and then get back to you.
You use this for drug delivery, but it's like stop somebody from like walking in the sun and what are we saying when they what you don't want to?
Yeah, so this technology is still like very, very far in development. So we haven't even really considered the implications of actually delivering it into someone. But there's lots of other problems that come with it. I mean, I would assume if you were to do that, maybe you'd do it in a place where there's minimal infrared radiation, because it's 800 nanometers. That is the desired absorbance of these nanoparticles. That specific wavelength is what excites it the most. And so I assume you would do that where that wavelength is minimally present. Plus, there's other problems you have just stuff in your body that will grab the nanoparticle and start to mash around it, and then it gets filtered out the liver. And so that would be a problem too, that if we were to use it later on. So.
Thank you, Dorian.